Thank you very much, Tony, for inviting me here. It's a great pleasure to give this talk in this honorable institution. Let me start right away. Oh, here it is, a little stronger. The problem in psychiatric diseases is that there's this is from a Nature paper a couple of years ago, the so-called drug deadlock, meaning that drugs don't really have new targets, but drug sales increase, and obviously not with more success than previously. And that is because psychiatric diseases, and I just talked about schizophrenia in this talk, are not very well defined in biological terms. Schizophrenia is a purely clinical diagnosis done in an interview. Underlying biological causes are heterogeneous, but they converge on a quite stereotypical reaction mode of the brain, which is psychosis or other rather unspecific symptoms. And the goal of what I'm going to tell you about and why I left clinical psychiatry to do fundamental research is to change that and to study the underpinnings of schizophrenia and eventually to change diagnostics and possibly introduce causative pharmacotherapies. Well, you know, probably all these operational definitions. This is from the former DSM-4. It's basically a checklist where you, as a clinical psychiatrist, go through a list of symptoms, and depending on whether you have enough checks, you eventually give the diagnosis of schizophrenia. And important, you have positive symptoms, you have negative symptoms, and a duration criterion, as well as excluding other disorders. And broadly speaking, this can be divided into positive, negative, and cognitive symptoms. Positive symptoms are the hallucinations, illusions, and so on. Negative are more unspecific, lack of drive, affective blunting, lack of personal care. And cognitive is very unspecific. It's more an attentional deficit, deficit in verbal, directive memory, and working memory. And while the genetics of familial genes has revealed candidate mechanisms, the study of common variants has not fulfilled all the expectations originally given, and explain at this point less than 10% of all cases. And this is also probably likely due to lack of stratification, meaning that you pool all those heterogeneous underpinnings into one clinical diagnosis. And what I'm going to talk about today is in this kind of hierarchical depiction of the different levels of biology, from genes to behavior. We're going to talk about this cellular level of disturbed cellular homeostasis or proteostasis. And this is not in contradiction to anything that is said about genes or even traumatic neurodevelopment. And obviously, what is also very important is that the criterion of non-adaptive behavior is very much also dependent on the norms and conventions that we give these clinical diagnoses. And a simple fact of postmitotic neurons is that the happy neuron can clear all the misfolded proteins that occur at all times by recognition and proteasomal or lysosomal degradation, whereas the dysfunctional neuron cannot do that because these metabolic and degradation mechanisms are impaired. And so you have a remainder of misfolded and aggregated proteins. And in the neurodegeneration diseases, this is very apparent through these microscopic aggregates that are visible even without further staining, like in Alzheimer's disease, the A-beta and tau aggregates, prion, the PRP plaques, and there are also smaller aggregates in trinucleate repeat diseases and Parkinson's disease. And so every disease has a kind of characteristic protein aggregate. And what is important is that the same proteins that are mutant in familial cases aggregate also in the sporadic cases. And that indicates that there is a high intrinsic propensity for the proteins to aggregate or misassemble, and that you could point to potential protein candidates when pedigrees with mutations have been identified. And it's really important to distinguish that you have in the neurodegenerative diseases these neurotoxic cell death causing aggregates, but there is a spectrum of protein aggregates where you have eventually just selective synaptotoxicity or even no toxicity at all, but some kind of dysfunctionality which impairs neuronal metabolism and leads to dysfunction in networking or action potential, forwarding, and so on. It's also very important to remember that many of those frontotemporal dementias initially start with psychiatric diagnoses. And that leads to the question whether maybe some of those cases could be frontotemporal dementias stalled at a very early stage. Don't get confused by this slide. This is the zoo of protein conformers and oligomers. In green are all those kind of that are soluble and functional. And a lot can happen if the protein is not functional. It can aggregate. It is usually degraded very fast. There are also some isolated cases where amyloid is functional. For example, in the posterior pituitary, hormones are stored in an amyloid form because it allows very dense packaging. But nature has arranged for a very nice de-amyloidation of those hormones when they're released. Our hypothesis, based on what I've just told you, was now that many chronic mental illnesses are lifelong diseases of the brain and have disturbed proteostasis. And so let's identify subtle protein aggregation also in these mental illnesses and use them as a biological phenotype. And we can, of course, do a hypothesis-free proteomics approach, and we can do a candidate approach. For doing this, basically what we do is we take postmortem brain collections. In this case was the Stanley Medical Research Institute's collection with 15 cases of both schizophrenia, bipolar disorder, unipolar depressive disorder, and normal controls. And we subject them through a series of ultracentrifugation steps, basically where we conserve the pellet. This includes high salts, high detergent conditions, and the remaining pellet is basically the protein aggregate. This was adapted from Gillian Bates' protocol for purifying Huntington's disease, Huntington protein aggregates. And I'm just shortly jump over the hypothesis-free proteomics approach, which is still ongoing. We've basically pooled two different brain collections obtained from Baltimore, the SMRI collection and the Liber brain collection. And we basically processed each case as I described, and then we validated them by single Western blots. And the interesting thing is we really got new candidates, which we are currently characterizing and hopefully publish quite soon. And I can't give you any details at this point, but rather I focus on this candidate protein approach. And again, as I told you, if you identify pedigree where mutations lead to the disease, the idea inspired by neurodegeneration research was that this could also be a candidate for a sporadic disease, in this case, schizophrenia. And the candidate was disrupted in schizophrenia 1, which is a gene identified in Edinburgh 20 years ago. And it's a large Scottish pedigree with cases carrying this translocation mutation leading to a haploinsufficiency phenotype and leading, and the carriers really have different diseases. It's so, even though it's called disrupted schizophrenia, they also have recurrent affective disorders depression, or even substance abuse disorders. And there was afterwards another family in the United States that had also a mutation leading to a shortened disc construct. Sorry. And by this logic, we probed the pellets of these purified cases. This is a Western blot for aggregated proteins. You see that in this lane, this is the control of the homogeneous input. And you see that there are only several cases and no controls. Here, S for schizophrenia, D for depression, B for bipolar disorder, and N for control. That there are only several cases that are clearly positive and much stronger in the pellet than the normal controls. And this kind of subset, about 15% of cases, were provisionally called dysguanopathies. And we didn't find those aggregates in neurodegeneration controls here, Parkinson's disease, Alzheimer's disease, DLB, and others. So they really appear to be very specific for schizophrenia. We also for 20 years tried to stain human brains. Now, this is a very bad slide of one of those stainings, and I have to say we haven't really succeeded in unequivocally showing that for two reasons. First is this is not a very highly expressed protein in the adult, and expression goes down in the adult. It's highest just after birth. And so in adult brains, you have only very small levels. Now, you could argue that aggregates would kind of stand out nevertheless, but there's a high background staining, also with lipofuscin, certainly these highly sensitive techniques. We haven't been successful to do this at this point. But what we did a couple of years ago is we used CSF from patients with first episode psychosis. And this study was done in collaboration with Marlene Pils and colleagues from the Department of Physical Biology in Düsseldorf by using a technique called surface-based fluorescence intensity distribution analysis. What's that? It's basically a sandwich ELISA where you have the same antibody coded and detected, and for detection labeled with two different fluorescent dyes. And the readout is not a biofluorescence reader, but by a laser who basically detects the single signals of these fluorescent dyes and kind of scans over these wells, which allows you to have a highly sensitive detection. And they developed this originally for Alzheimer's disease. And you can use a standard curve by basically cross-linking silicon nanoparticles to the epitope of the peptide used. And this is such a standard curve, and you see these single points. These are the aggregates detected by the laser, and you have pico or attomolar even concentrations that you can detect. And you have a nice standard curve that you can then calibrate and measure the concentrations. And by doing this, and we used this in collaboration with Sophie Erhardt from the Karolinska Institute, who has a nice collection of 50 CSF samples from first episode patients, versus matched healthy controls. And the average concentration of disc aggregates in this assay was higher, and particularly standing out again, a couple of cases, which are probably the interesting cases where we find higher disc aggregates in these CSF samples than in the normal controls. And these higher samples, interestingly, they were caused, if you do a clinical subset analysis, by a subset of seasonal affective disorder and undefined schizophrenia. So it's this kind of borderline diagnostic spectrum with affective disorders and atypical cases that seem to be particularly associated to disc aggregates or to disc-related pathology. And also a genetic polymorphism segregated with these aggregates. Now, back to fundamental research. Disrupted in schizophrenia has a high aggregation propensity, particularly in a C-terminal region. And has many characteristics that others have also to be true for aggregates in the neurodegenerative diseases, such as cell invasiveness. And we show together with Chiara Storzolo that these disc aggregates in vitro can transfer into other cells and kind of their seed aggregation. And when we injected a recombinant purified protein that is very aggregation prone, we also could show that it is readily taken up into brain cells of an injected rat. Basically, the way we see it is that at disk, this is a AlphaFold2 structure. There is no structure for a disk yet. I think there is a publication underway that eventually will address that. Very low concentrations, such as in the Scottish family, lead to a haploinsufficiency phenotype, which causes mental illness, and very high concentrations lead to aggregation and to loss and gain of function, leading also to aberrant disk signaling. So there is a narrow window when a disk will work properly. And in order to work properly, this is from papers from a couple of years ago, just showing that disk interacts with 200 proteins or more, that it is really at the crossroads of many signaling pathways, meaning that when it is impaired, you influence really a lot of different functions. Now, to model this in animals, we generated transgenic disk rat, and the advantage of the rat versus the mouse is even though we can crossbreed it with other mutations, it's better for imaging, better for behavior and anatomical studies, and improves also biomarker collection. For example, you can easily take CSF. And when we generated this rat 10 years ago, we saw that indeed we could mimic these aggregates, and was modestly overexpressed like 5 times. So we choose to study only those, this red line that had a very modest overexpression in order not to run into too many artifacts here. And we had solid expression of aggregation. And interestingly, the aggregates were strongest in those regions of the brain that are high in dopamine content, such as the dorsal striatum. Interestingly, we did not see amyloid here visualized by THS staining. We did see amyloid when we expressed this in E. coli, but not when overexpressed in this transgenic disk rat. And this is part of the collaboration that we did with Tony. Initially, many years ago, we saw slightly elevated ventricles, and in a very new study published last year, we also saw connectivity differences by diffusion tensor imaging. This is a study done by Josie Duran and Julia Koparnikova from Düsseldorf. We saw different connectivity in different regions relevant for social behavior, as I will explain in a second. We initially focused very much on this dopamine metabolism because it's a prominent feature in schizophrenia and psychosis. And what we saw is the animal at 4 and 20 months overexpressing disk and leading to these disk aggregates had hypersensitivity to amphetamine. This is a phenotype that's also seen in patients. So a dosage that in wild-type controls would not lead to hyperlocomotion. This is a preferred readout for this phenotype in rats or other rodents. Would in the transgenic disk rat lead to hyperlocomotion, meaning that there is a increased triggering of protopaminergic pathways. Together with Philip Seaman from Toronto, we then analyzed the dopamine 2 receptors, and they found that there were increased high affinity dopamine receptors, and in this scale that he has put up, we would be in the lower part of that high affinity status. And we also saw interactions with dopamine transporter. We saw physical interaction by immunoprecipitation and co-staining here. also by cyclic voltammetry, where the clearance in the transgenic disc animals is impaired. And anatomically, a student from me, Hannah Hamburg, also investigated the dopamine system here with the TH tyrosine hydroxylase staining, and we saw that the number of substantia nigra neurons was decreased, as well as the projections to the striatum, leading to an increase of striosomes. In parallel, apparently, also interneuron architecture changed with a decrease in the upper layer interneurons and an increase in the lower layer interneurons in this rat. So apparently these dopaminergic and interneuron developments kind of are inversely related and governed by disc protein. Some other groups also did more complicated studies. One was the group of Joseph Chichwari in Vienna, who did some hippocampal recordings leading to his claims of smaller place fields in the transgenic disc rat and altered spatial coding, as well as differences in theta wavelength, sleep, and since it's not my own research, I'm and not so familiar with it, and so you can read this in these papers that are indicated here. And we also gave this rat to a company from Canada, Transformation, who did some sleep EEG measurements. You can see the link here. And what you see is you have decrease in both theta, delta, and sigma wavelength in this transgenic disc rat. And these are the most recent studies done by Jose Duran from the Department of Social Psychology, who now joined my lab. One is a very interesting assay. It's called social transmission of food preference. This is an assay that is investigating social learning of rats. And the way it's done is you basically have one rat eating a preferred choice of either banana or berry. And for example, if the rat then picks berry as a preferred taste, you match it with a rat that eats banana preferred, and then you put them together, and then the older rat smells the banana and then eventually shifts its preference from berry to banana as kind of a social learning, and it makes evolutionary sense to eat something that somebody else has already eaten, because it's safer to choose food that has not impaired the other part. And what you see is that the wild type rat would change to a significant degree the preference for that food, but the transgenic disc rat doesn't. It meaning that there is some communication deficit in the transgenic disc rat, and this was compatible with the regions that were where we saw these connectivity differences in the rat that have been described to code for these social functions. In another paper published this year, we did a related test, which was called social novelty, where one measures basically the degree of interest that a novel rat introduced into a cage where the animal to be tested has the choice between an older contact rat and a novel rat, would spend more time with the novel rat as an indicator of social interest, and the transgenic disc rat did not show that social interest. This is shown here. There's no difference here, but in the wild type difference, there is this difference. And this difference could be rescued by administration of amisulpride, a D2, D3 dopamine 2 receptor inhibitor. And interestingly, in the wild type rats, this led to the disappearance of this preference, meaning that there's clearly a difference in the physiology of these two rats. We also did some immunological investigations, and to begin with, we analyzed the PBMCs from the blood, and what we saw is the transgenic disc rat had a decrease or modulation in a distinct network of genes characterized by RGS1, CCL4, and NKG7. These are genes overrepresented in T lymphocytes and macrophages, and the analysis was done via this weighted network analysis done by Ovidio Popa from the Institute of Bioinformatics at the University of Düsseldorf. And when we tested these three proteins in two different human subsets, sets of patients with schizophrenia. One was from Düsseldorf Psychiatric Clinic, one was from the clinic from Peter Falka in Munich. We saw that these patients also had decreases in the same transcripts isolated from PBMCs as the transgenic disc rat, meaning that really this seems to be a relevant network. And if you then put those three genes together, you can arrive at a diagnostic specificity with 97% and a sensitivity of 31%, indicating that it's a very specific subset. It would not recognize all the cases of schizophrenia, only this subset. It's a matter of stratification. Now you may ask, oh, well, you're overexpressing this disc gene in this rat, and this leads to a lot of artifacts. And is there also disc protein aggregation without a transgene overexpression? I'll show you two examples. One is by viral infection, and the other is by oxidative dopamine originating stress. So, and this also relates to another strain of research that we are doing in Düsseldorf, which is how is protein aggregation caused by virus infection. And you must know that viruses, when they replicate, they use the cellular machinery. They mess it up basically by hijacking all the functions to their own better replication. And by doing that, they have spotted all the weak spots of the cell. And since they evolve much faster than the host cell. And what we have done in this experiment that I'm going to show you is we have taken the influenza H1N1 strain, which was adapted to mice, and the original strain, and we infected both human Loomis cells, these are primary-like cells, and rag knockout mice. Why rag knockout? Because the rag knockout is a cell without a functioning adaptive immune response, allowing the influenza viruses to enter the brain more easily. And we published this 5 years ago. Here are the cellular experiments. What you see here is these Loomis cells. No infection leads to, and we analyzed several protein aggregates candidates, synuclein, disc, tau, and TDP43. And I can already tell you that for tau and TDP43, we didn't see anything. But for synuclein, we did see the induction of endogenous aggregates in these primary-like human cells. And we also saw that we also saw a couple of, it's very small here, it's much smaller than the synuclein aggregates, a couple of induced disc aggregates. And when we looked in these influenza-infected rag knockout mice, we saw that also here, the synuclein was highly upregulated, forming aggregates. And the same was true for disc, but yes, as you can see here. But it was interesting to see that these synuclein aggregates would not co-aggregate with disc. Sometimes you have co-aggregation, but apparently for disc and synuclein, this is not the case. What you see here is the green synuclein aggregates and much smaller disc aggregates. And we believe this is because the synuclein, the influenza virus both influences the autophagosome generation as well as the fusion of lysosomes with autophagosomes. And we're continuing research on this, but I don't have time to show you about this, more about that. What I want to stress is that these are pathways where you can, when you modulate them or inhibit them, you will eventually cause physiological endogenous disc aggregates. Another thing that is very reliable is the induction of disc aggregosomes by dopamine. So you just add 100 nanomolar, millimolar of dopamine into cell cultures, and you can cause these disc aggregates, and they are transient because if you take dopamine off, you decrease them. This is a electron microscopical stain, and these form then vimentin-coated aggregates that co-localize with the proteasome. And another experiment in the same line was with, this time with wild-type Wistar rats. In this case, we didn't use a one-shot amphetamine sensitization reaction testing, but a true sensitization. A true sensitization is basically when you apply amphetamine for 5 days, and so to deplete the dopamine neurotransmitter storage capacity, then do a 14-day pause, and then test again with one shot of a lower dose of amphetamine. And when you do this in wild-type rats, what you see here is you have, so this is saline, this is the basic condition, and when you look at the stratum, again, this is what I showed you before, you have more disc aggregation in the dopamine-rich tissue regions in the brain, whereas in the medial prefrontal cortex, you have less dopamine and accordingly less dopamine aggregates. After depletion of dopamine by this procedure, you also have less aggregates in the stratum, but you have a slight increase in dopamine in disc aggregates in the prefrontal cortex. The difference is significant in this stratum. And this is also shown here, this inverse correlation of the aggregates and the behavioral testing for locomotion. So dopamine is another stressor that can cause endogenous disc aggregates. And I told you before about this. Now we're getting back again to the one-shot amphetamine supersensitivity. And we published this 10 years ago, and then we tried to replicate it many times, and we weren't able to replicate it. And so we were asking us what happens. And other groups have received this RAD2. This is a group in Erlangen, Christian Müller, I think he was here in this institute many years ago, and a Brazilian group, and they were also able to see this phenotype. But again, we had problems, and also this Canadian company had problems. And we're currently investigating what the reason is, but it may be very interesting, because the question is whether it is a metastable phenotype that eventually mimics cyclical psychosis. So if you take amphetamine sensitization or hypersensitivity as a proxy for psychosis, the modulation of it may indicate under which conditions patients could eventually have increased risk for psychosis. And what is well known is that amphetamine sensitivity is augmented by prior neuroinflammatory activation and by the sex. So, for example, females have a higher propensity than males, and this has been published by Christian Müller's group. And now, what our current model is that amphetamine supersensitivity is this metastable phenotype that is governed by variable levels of disk aggregates. And what we see in this red, and this is unpublished data, is we have decreased IL-10 levels. IL-10 is an anti-inflammatory cytokine, and it has been described that amphetamine basically acts via the astrocyte and IL-10 to inhibit microglia activation. And what we show, what our idea is that David Porter's group in Edinburgh had shown that DISC and Housley from Glasgow, that DISC inhibits PDE4B, which then increases CAMP1 levels, which increase IL-10 levels, and they suppress neuroinflammation. But if you have DISC aggregates, this inhibition may be stalled, so that in fact you can have you can decrease the CAMP levels, the IL-10 levels, which leads to increased neuroinflammation. And by basically modulating the DISC aggregates, you may have this variability. It's a kind of a stress sensor that may correlate with psychotic occurrence. So, summary of the transgenic DISC rat. We have aberrant dopamine homeostasis shown by neuroanatomy, differential interactions of DISC with that lead to high affinity receptors, and others, subtle neuroimmunological changes which are currently not fully characterized, like this PBMC signature, lower IL-10 levels, and behavioral changes. And for example, if we now plot this onto the symptom dimensions of schizophrenia, we have the amphetamine supersensitivity for positive symptoms. We have these negative symptoms, social deficits, and we have cognitive symptoms like these memory consolidation deficits. I didn't talk about the novel object recognition at 24 hours, which was also impaired in published from the Joe Houston's lab. And so these transgenic DISC rat phenotypes are comparable to all aspects of schizophrenia, of the schizophrenia symptom spectrum. And going full circle, we really believe that I've shown you that we can define a subset in postmodern brains. We can model this in this DISC rat. We can develop biomarkers like these DISC aggregates in CSF or this PBMC signature. We can even do tailored pharmacology, which I showed you with this amisulpride rescue of the social deficit phenotype, and something similar has also been described in patients with amisulpride being beneficial for social phenotypes. Conclusion: the transgenic DISC rat reflects features of a subset of sporadic cases. It has high face validity and predictive validity, and on a molecular mechanistic level, DISC1 misassembly seems to modulate dopamine homeostasis, and I've called this subset DISC1opathies. And certainly, the transgenic DISC rat is useful to model a subset of sporadic chronic mental illnesses. Take-home message: subset identification is key for advancing molecular research, and aberrant proteostasis is an interesting phenotype to achieve such subset stratification, and our approach is complementary but not exclusive to other approaches. Tony asked me to just mention that we have more research on this virus and drug discovery. And if you're interested, we can talk about this afterwards or in a discussion. Thank you very much. I mentioned all my collaborative workers that participate in this research. So here they are again, and thank you very much for your attention.